now we have been reading about the principles and processes of biotechnology. In the principles and processes of biotechnology, you have primarily understood the definition of biotechnology. You had learnt about the sort of tools in biotechnology, especially restriction enzymes and cloning vectors. Later on, you knew about how do you isolate a particular sort of a gene, how do you select your sort of a transferments. You had also learnt about PCR, polymerase chain reaction, and knew about how do you have a competent host, and which are again methods by means of which your foreign gene of interest should be, how it should be inserted into the sort of a host cell. Now, parallel to the principles and processes in biotechnology, we have the second unit now. The second unit now is termed as biotechnology and its applications. Has a link to chapter 1 and chapter 2. You would be learning initially about a small vector which is termed as Agrobacterium tumefaciens. Now, Agrobacterium tumefaciens is normally termed as a natural genetic engineer. It is a soil negative bacteria which is grown in tropical soils associated with the roots of dicots, monocots and gymnosperms. If you look at nature, in nature we have Agrobacterium tumefaciens which acts as a sort of a natural genetic engineer, that is, we do not have to do anything about it. The plant or the bacteria all by itself, which is a soil negative bacteria present in tropical soils associated with the roots of dicots, monocots and gymnosperms, all by itself can do the process of genetic engineering. So, it is God's gift to genetic engineering of biotechnology. So, what could Agrobacterium tumefaciens do? It can transfer its own DNA naturally into plants. So, it has got a capability of transferring its own DNA. Agrobacterium has got its DNA. This DNA is gifted to plants. It can transfer it to primarily into plants. And Agrobacterium tumefaciens is a very important sort of a pathogen. It is a pathogen which is responsible for a disease which is termed as crown gall disease. Crown gall disease is a bacterial canker. It is a cancerous disease. So, it causes plant cancer. It causes plant cancer. Thereby, in view, because it can cause a sort of a crown gall disease, its DNA can spread very fast. In the host cells, it can spread very fast in the host cells. And again, when you take the Agrobacterium tumefaciens, when you take its sort of a DNA, the DNA can regulate. The DNA can regulate plant growth and development. It can totally take control of the plant growth and development. And at the same time, the DNA is also responsible for the synthesis of Agrobacterium tumefaciens is also responsible for the synthesis of auxins, which are growth hormones, cytokinins, which help in division of the cells or cell division, and a group of compounds which are termed as opines. So, the Agrobacterium tumefaciens as a candidate is a very important source of is a natural genetic engineer. And the Agrobacterium tumefaciens is a bacteria which has its own plasmid, and the plasmid which is present in Agrobacterium tumefaciens is termed as tumor inducing plasmid. It is popularly designated as TI plasmid. The TI plasmid has got the oncogene. The TI plasmid has got the oncogene or the disease causing, it's the cancerous disease causing gene. Now, when you take the TI plasmid, if you think this is the TI plasmid, the TI plasmid will have a region which is called the tDNE. We are talking about TI plasmid. Plasmids are circular molecules, and the TI plasmid has got a region which is termed as a tDNE. Now, the tDNE, when you take the entire sort of a tDNE, it has got a right flank and it has got a left flank. The tDNE has got a right flank and it has a left flank. The advantage over here is the foreign gene of interest, your foreign gene of interest can naturally be inserted between the left flank and the right flank. So, when a foreign gene is inserted, when you have a foreign gene which is inserted in the tDNE region between the right and the left flank, the TI plasmid gets inactivated. So, your foreign gene acts like a sort of an off button for TI plasmid. It gets inactivated or your TI plasmid gets aborted, or you can say it becomes inactive. So, when it becomes aborted or inactive, the reason is there is deletion, it undergoes deletion. There is deletion of the oncogene. The place where the cancerous gene is present, that is cut off, and in that particular place your foreign gene of interest comes in over here. Now, once you have your foreign gene which is linked in the region of the tDNE where supposed to be you have your TI plasmid, then the transfer of the foreign gene along with the tDNE in the TI plasmid is very, very efficient. So, thereby what are the advantages? Why do you call Agrobacterium tumefaciens as a natural genetic engineer? Because it has the sort of a TI plasmid. Next, the TI plasmid is disarmed. Third one, the foreign gene of interest is placed into it. It has got a foreign gene insert. Then it has the tDNA which is functional. The tDNA is functional. Thereby, it can cause efficient transfer of the foreign gene into host cells. So, we are talking in terms of efficiency. So, it is an efficient gene transfer vector. It is an efficient gene transfer vector. Number one is that, and next one is it can directly take your sort of a foreign DNA of insert and it can integrate it with the plant DNA. So, imagine that we have a TI plasmid. From the TI plasmid, we have the tDNA. In the tDNA, the gene, the oncogene which is very important for crown gall is knocked off, and your foreign gene of interest is placed in the region of the tDNA. The tDNA from the bacteria is uploaded into the sort of a plant DNA. It integrates with the plant DNA, and thereby we have an efficient method of transfer of the sort of a host cells of the plant DNA. So, you can primarily, Agrobacterium tumefaciens is very efficient in terms of genetically engineering crops, more so with relation to developing transgenic brassicas. If you take your cabbages, cauliflowers, carrots, all of these belong to Brassicaceae, even including mustard. So, genetic modification of Brassicaceae or cruciferae, which includes cabbage, cauliflower, carrot, they can be primarily be done by means of Agrobacterium tumefaciens. Now, the tumor inducing plasmid in Agrobacterium is the most important plasmid which has got a very important role with relation to plant genetic engineering. So, 80% of plant genetic engineering which occurs in the world occurs by means of Agrobacterium tumefaciens, and the most important valid plasmid for genetic engineering is the TI plasmid. So, what exactly is the TI plasmid? The TI plasmid is a large circular DNA. It is a large circular double stranded DNA, and it has got a sort of a left border. It has got a left border and a right border, or a right flank and a left flank, and it also has a sort of a gene called VIR. It has got a gene called VIR. VIR indicates virulence. So, it always carries a gene for virulence, so that it can attack the sort of host cells. Now, here again the virulence gene can also be removed. The virulence gene can also be removed, and your foreign gene of interest can be inserted in place of the sort of a virulent gene. So, you can take the oncogene out, or you can also take even the sort of a virulent genes out. So, whenever we take a sort of an oncogene out or your virulent gene out, you call this sort of a vector as a disarmed vector. So, what is the meaning of the term disarmed vector? A disarmed vector is one in which the virulence gene or else the oncogene is removed, and in place you got the foreign gene of interest. Now, what exactly are the advantages of Agrobacterium tumefaciens? The advantages of Agrobacterium tumefaciens is it shows high frequency genetic transformation. It means the number of the cells which have been genetically transformed are very high. High frequency genetic frequency is possible by means of Agrobacterium tumefaciens, and most of the transformations or most of the cases where the foreign gene has been inserted, or you have a recombinant DNA which is carried by means of the TNA plasmid, most of the transformants are highly stable. They are stable and they are inherited. Now, stability in the case of plant biotechnology is very important, because the cells should not suffer the shock of taking a foreign gene. The cells are very much stable and they show normal metabolism. And another very important point is Agrobacterium tumefaciens can always take a large DNA insert. It is just like a vehicle which carries a lot of passengers. So, it can carry about the greater than 150 kilo base pairs of DNA. So, a large chip of the DNA can easily be transferred, and again it has got a wide host range. Because it is naturally present in dicots, it has got a wide host range of dicots. And TI plasmid is a very important plasmid, and the TI plasmid is a resident plasmid. It is termed as a resident plasmid. That is, the tumor inducing plasmid is always associated with the Agrobacterium tumefaciens. The plasmid is always piggybacking on Agrobacterium tumefaciens, and the method of transfer is termed as a natural gene transfer. Now, when you take Agrobacterium tumefaciens with your TA plasmid, a very important point is it serves as a binary vector. A binary vector is one which can move from one plant to another plant, one plant to a bacteria, from a prokaryote to a eukaryote, and it is capable of expression in 2 cells. So, it is termed as a binary vector because it can easily shuttle between a plant and a bacteria. It can carry the genes and it is also capable of expression, gene expression. The true test whether biotechnology is successful or no is, after the foreign gene has been transferred into the plant, can the DNA again make the RNA and the proteins, and can it go to form a true biosynthetic pathway as it occurs in natural systems. So, gene expression is a very important test. So, the advantages of Agrobacterium tumefaciens is it shows high frequency gene transfer, it is stable, it is inherited, it has got a large DNA insert, it has got a wide host range. Then you have if you take the TA plasmid, it is a resident plasmid. It is also a sort of a binary vector. It is called a shuttle vector, and it can also express in bacteria and also in the case of plants. So, these are the advantages of the Agrobacterium tumefaciens.